हॅलो रणजित, आवाज येतोय? मला येत नाही तुमचा आवाज.
हा आता येतोय का सर बघा?
हा येतोय येतोय येतोय.
हा 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 ते म्यूट केलं होतं मी, तर आम्ही बोलत होतो त्याच्यामुळे.
बर बर.
हा 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 आणि ऍड केले सर ते आता बघायला. प्रत्येक आता जवळपास 66 जण आलेत ऑनलाईन.
बर बर बर, चालेल
हा
चालेल.
हा हा हा
माझ्याकडचा दिसतोय तुम्हाला कॅमेरा? कॅमेरा ऑन आहे?
तुमचा व्हिडिओ नाही दिसत सर आणखीन.
आवाज येतोय रणजित?
हा आवाज येतो सर क्लिअर, व्हिडिओ दिसत नाही.
तुमचा येत नाही मला आवाज.
आवाज नाही येत का? आता बघा सर, येतोय का?
तुमचा आवाज नाही येते.
हॅलो, येतो का सर बघा आता? हॅलो हॅलो
हॅलो.
हा आता येतो का सर बघा आवाज?
नाही नाही, आवाजही नाही आणि माझा व्हिडिओ बंद आहे तुमच्याकडनं.
माझं नाही, माझ्याकडे नसतं बंद
माझ्याकडनं
सर.
व्हिडिओ चालू आहे.
चालू आहे का?
दोन्ही चालू आहे माझ्याकडनं.
मग काय प्रॉब्लेम झाला इथे? तुमचा व्हिडिओ चालू आहे सर इथे.
नाही,
तुमचा व्हिडिओ दिसतोय मला इथे ऑन, म्हणजे ब्लॉक नाही केलेला तो.
व्हिडिओचा ऑप्शन चालू आहे.
हा ऑप्शन चालू आहे का? एक मिनिट. चालू आहे सर. तुमचा व्हिडिओ दिसत नाहीये.
हॅलो.
हा व्हिडिओ दिसत नाही म्हटलं, तुमचा आवाज येतोय मला.
आवाज येत नाही मला.
हा आता येतोय का सर बघा? हॅलो हॅलो हॅलो
हॅलो.
हा आवाज येतो का सर आता? हॅलो हॅलो. 91 जण झाले आता. नाईकवाडे सर कुठे गेले?
यामध्ये कॉमन सब्जेक्ट आणि स्पेशल दोन हँड आणि मानकांना स्वतः कमी किमतीत मिळून जाते.
आवाज नाही होते.
किंमत किती आहे अजून मला कळलेली नाही.
हॅलो सर, तुमच्याकडनं बहुतेक ते म्यूट झालं असेल. हॅलो हॅलो सर.
हॅलो.
हा आवाज येतो सर तुमचा मला.
हॅलो.
हा तुमचा आवाज येतो सर मला.
रणजित.
मॉर्निंग सर. हॅलो.
हॅलो.
आवाज येतोय?
आवाज येतोय का?
तुला आवाज येतोय? त्याला येतोय हॅलो. आपला तर आता आवाज यायला लागला. हॅलो.
हॅलो.
हा आवाज येतोय?
हॅलो.
हा रणजित, आवाज येतोय का?
पण मला येत नाही आवाज.
हा हॅलो सर बोला.
आवाज येतोय का म्हणलं?
हा येतो सर निखिल सर.
हा सर माझा आवाज तर येतोय.
तुमचं हे
काय
बघा,
प्रॉब्लेम झाला?
स्पीकर बघा, तुमचं कनेक्शनचा प्रॉब्लेम असेल तुमचा.
हा सर तुमचा आवाज येतोय.
बर बर बर.
हा गुड आफ्टरनून सर.
हा येतो सर, येतोय आता.
ओके ओके. ऑडिबल
नाही, ऑडिबल नाही आणखीन.
माझ्याकडनं काही प्रॉब्लेम नाही.
मला काय येत नाही आवाज मग.
हॅलो.
हा
हा,
हॅलो,
हॅलो.
बोला बोला सर, बोला.
आवाज येतोय?
माझ्या ह्याला काही नाही.
हा सर येतोय तुमचा आवाज.
दुसऱ्या सिस्टमला आवाज येतोय?
हा येतोय, तुमच्या येतो सर आवाज.
आपल्या सिस्टमला ओके.
येतोय सर.
हॅलो.
हॅलो.
हा येतो का सर आवाज आता?
येतोय येतोय.
हा येतोय ना आता?
हो येतोय. तुम्हाला
हा
येतोय?
येतोय. हा मला पण येतोय सर, आता मला पण येतोय.
ओके
हा
ओके.
ओके सर, ओके ओके. हा हा, याच्यात प्रॉब्लेम झाला असेल.
एक काम करा. हॅलो.
हा सर बोला.
सरांना स्क्रीन शेअरिंगचं ऑप्शन ऑन करून द्या. सध्या तुम्ही बंद केलंय.
स्क्रीन शेअरचा ऑप्शन ऑन करून ठेवतो.
सरांसाठी फक्त.
इथं जा, हे करायचं ना.
ऑल करा. ऑल करा. नाही ते नको. खाली ॲडव्हान्स. ऑल पार्टिसिपेंट.
ऑल पार्टिसिपंट्सला नका करू. ऑल पार्टिसिपंट नका करू, फक्त मला करा. कारण नाही तर मग काय होतं, मध्ये स्ट्रीमिंग लोक काहीतरी
हा हा, दुसरे देतात काय? ओके ओके.
हा हा, फक्त मला करा त्याच्यामध्ये स्क्रीन शेअरिंग साठी.
एक मिनिट करतोय सर आधी.
हो हो. म्हणजे स्क्रीन दिसते का तुम्हाला?
हा दिसते दिसते सर, दिसते.
ओके 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 डन. ओके चालेल
हा हा
चालेल.
हा हा. हा दिसते सर स्क्रीन आता.
हा हा
हा
चालेल
हा
चालेल.
हा.
ओके. लावू मी हे?
पंच्याण्णव. तुम्ही पण मला वाटलं तुम्ही शोधतोय तुम्हाला ह्याच्यामध्ये मी. हा हा. हा हा.
अमित काय म्हणताय?
वरळे सर, सर बोलताय तुम्हाला. हा.
हा हॅलो.
बोला बोला सर.
हॅलो सर.
हा अमित. हा सांगा, कसे आहात?
हॅलो. हा
हॅलो.
बरं आहे सर, तुम्ही कसं आहे?
हो छान आहे, छान. पप्पा कसे आहेत?
हा बरे आहेत सर, बरे आहेत.
कुठं निपाणीला आहेत की काय इकडे आहेत मुंबईला?
पप्पा इकडे बहिणीकडे गेलेत नेरूळला सर.
बर बर बर. अच्छा.
हा काका कसे आहेत?
आहे व्यवस्थित आहे.
हा काकाने सांगा जय भीम.
हो हो.
हा
ओके.
हा, काळजी घ्या सर. हा.
हा मॅडम, तुम्हाला थोड्या वेळात सांगतो, मीटिंग मध्ये आहे मी. मी बघितलं नाही हेडफोननी काय. अच्छा, तो सोनीचा स्पीकर आहे ते, पण त्याला हे नाहीये. तो नाही असू दे. अरे जरा सरळ बोलू बरं. काय बंद काय झाली परत? रणजित, आवाज येतोय माझा?
हा सर, येतो आवाज सर.
हा, आवाज येतोय सर तुमचा.
बरं.
हा, येतोय आवाज. आम्ही जरा बोलत होतो त्याच्यामुळे मी म्यूट करून ठेवलं होतं माझं.
अच्छा, बरं.
हा 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 हा.
माझा व्हिडिओ बंद केलाय काय?
हॅलो, चालूच आहे सर तुमचा व्हिडिओ.
ते मला दिसत नाही, म्हणजे हे दिसत नाही.
तुम्हाला दिसत नाही का?
हा हा हा.
नाही, आम्हाला दिसतोय तुमचा व्हिडिओ. ते जन असरायचं.
सरांना बोलूस. आहे, माझा व्हिडिओ बंद झालाय. माझा व्हिडिओ कधी दिसत नाही? रिटर्न टू मीटिंग. आता हॅलो.
हा, बोला बोला सर.
आवाज येतोय? आवाज
हो, येतोय आवाज. आवाज येतोय.
स्क्रीन दिसते?
स्क्रीन पण दिसते.
अच्छा, सर दिसतायत?
हो, सर पण दिसतायत. फक्त जरा कॅमेरा खाली करायला पाहिजे थोडासा.
एक मिनिट.
हा हा, व्हेरी गुड. हा, दिसते आता.
ओके.
हा हा, दिसते सर.
नाही हे जे आहे ना एक हाईड करून ठेवलंय बाकी हे सगळं चालूच आहे ऑलरेडी हे चालूच आहे. एक मिनिट. हा, आता प्रेझेंटेशन आणि सर सगळे विषय दिसतात. एक मिनिट.
हा प्रेझेंटेशन दिसतंय लावलेलं रोल ऑफ इंड्यूस म्युटेशनचं आणि सर पण दिसत आहेत.
ओके ओके
हा हा हा
ओके.
हा नाही याला काही करत नाही फक्त हे आपलं स्लाइड शो बस एवढंच. सुरू केलंय.
बाकी अडचण नाही आहे. इंटरनेट वगैरे ओके. अरे सुरेश,
Nikhil sir, good morning.
मॉर्निंग.
Good morning. Good morning. Pansode sir. हे असं काय झालंय? नाव यू आर अलाविंग देम काय असं झालंय?
हा सर हा सर
अच्छा 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 अच्छा.
हा आता सगळ्यांना अलाव केलं सर मी त्याच्यामध्ये.
Okay okay okay.
हा हा सर.
I was just waiting to increase the number, so I thought.
हो सर.
Yeah. So in all 95 are there online.
येस सर.
I think we can start.
येस सर येस सर.
Will it be all right?
Yeah.
Nikhil sir, you are okay?
Yes sir, yes. I'm okay sir.
Okay.
शॅल आय स्टार्ट सर?
हा वेलकम मी करू ना?
मी आधी बोलतो नंतर सर करा.
बरं ठीक
हा,
आहे
हा,
ठीक आहे
okay.
ठीक आहे ठीक आहे.
Respected resource person, Dr. Nikhil Gaikwad sir, Professor, Department of Botany, Shivaji University, Kolhapur. Respected principal and convener of this national webinar, Dr. Narendra Tendulkar sir, all the participants from various states of India, and my dear colleagues and student friends. I am Dr. Ranjit Bansode. I welcome you all on behalf of Devruk Shikshan Prasarak Mandals, Atle Sapre and Pitre, Autonomous College, Devruk. I request Honorable Principal Sir, please give best wishes for this webinar. Sir, सर आवाज थोडासा कट होतोय.
हॅलो.
हा सर येतो बोला आता.
Audible?
हा यस सर यस सर.
Hello, good morning.
Morning sir.
I'm in resource person for the today's national level webinar, Dr. Nikhil Gaikwad, Head Department of Botany, Atle Sapre Pitre College, Dr. Pratap Naikwade, my professor colleague, Dr. Ranjit Bansode, professors from the various colleges, researchers, students, I extend very warm welcome for the one-day national webinar, Role of Induced Mutation in Crop Improvement and Crop Production. Sir, it's my special thanks to you. You have consented, you have accepted. So on behalf of Atle Sapre Pitre College and Devruk Shikshan Prasarak Mandal, and in fact on behalf of all participants, I thank you all. Friends, in the past several decades, induced mutations have contributed immensely to the development of improved varieties of several crop plants. Induced mutations have played significant role in meeting challenges related to world food and nutritional securities. Very recent statistics says that more than 60% of officially released mutant varieties are from Asia. With China, India, Japan, they are topping in the list. Actually, the genetic variations is a mainstay to produce new and improved cultivars. Evolution and practical breeding depend on genetic variation is actually the known fact. Genetics is actually the fundamental science of plant breeding. I am sure our resource person for the today's webinar, Dr. Nikhil, will definitely address various aspects of mutation induction, like how do plants induce mutations, how do plant breeders use mutation to improve crops, mutation applications, and examples of successful use of induced mutant in crop improvement. I think without taking much time, I would request Dr. Nikhil to proceed. Dr. Nikhil, please you take on. Thank you once again. God bless you all.
Thank you, sir. Introduction given आहे. It gives me immense pleasure to introduce the resource person of this national webinar, Dr. Nikhil Gaikwad, sir. He has completed MSc, NED, PhD, and working as a professor in Department of Botany, and he also has a Management Council member of Shivaji University, Kolhapur. He has 22 years of research and 18 years of PG teaching experience. Under his able guidance, 1 MPhil and 6 PhD students were awarded the degree. Currently, 4 students are working. He has completed 5 major and 2 minor projects funded by UGC, MOEF, DPDC, RGSTS, and it has a worth about 2.6 crore as a principal investigator. He also has 2 major ongoing projects funded by Department of Atomic Energy and DST as a principal investigator and worth about 70 lakh rupees. He has published 53 papers in various national and international reputed journals of biological sciences. He has presented his findings in 69 national and international conferences. He also recorded Crotalaria srirangiana, member of family Fabaceae, as a new record from the Western Ghats of India. He elected as a Senate member from PG teacher constituency of the Shivaji University, Kolhapur from November 2017. He elected as a management council or syndicate member of Shivaji University, Kolhapur from February 2018. He working and worked as secretary for SUPTA. He also working as a member of grievance committee of Shivaji University, Kolhapur from February 2018. And he also working as a sectional member of botanical section of Indian Science Congress Association from Western India region from 2020 to 21. He also visited International Atomic Energy Association, Vienna, for International Symposium of Plant Mutation Breeding and Biotechnology. And this conference was organized by Food and Agricultural Organization of the United Nations in August 2018. So such a eminent personality we have as a resource person for this national webinar on role of induced mutation in crop improvement and crop protection. I request Dr. Nikhil Gaikwad, sir, please enlighten the delegates.
Okay, thank you.
Okay, sir.
Good afternoon to all of you. Am I audible?
Yes, sir. Yes, sir.
Good afternoon to all of you. Devruk Shikshan Prasarak Mandals, Autonomous College Principal Dr. Tenulkar sir, head and convener of this particular webinar, coordinator and head of the department of college Dr. Pratap Naikwade, organizing secretary Dr. Ranjit Bansode, and all the faculty members of college and participants who have joined for this particular webinar. So in this particular webinar, I would like to share some of my views regarding mutation. So in which role of induced mutation in crop improvement and crop protection. So that is the particular topic in which first half, what I will do, I will try to share what are the particular steps involved in induction of mutation, then how mutation can be used for protection of crop, and last part I will share my experiences, self experiences in from field. So how mutations are useful for creation of genetic variability and so on. So we'll start first half part one by one. As we know, mutation. So mutation term was coined by Hugo de Vries in 1900. where we can see that particular change in genetic constitution of an organism that must be a heritable one, and that is induced by physical or chemical reaction of the agent, and that is not because of segregation or genetic recombination. So such kind of changes are called as your particular mutation. Now, if you see that particular types of mutation, there are two types of mutation, broad sensely. So spontaneous mutation and induced mutation. So spontaneous mutations are those type of mutation which are induced naturally, and their rate is very, very low, 1 into 10 raised to minus 7. So that much less rate is there. As compared to spontaneous mutation, induced mutations are much more, are having much more higher rate, where we can observe a change in the genetic constitution of an organism because of either action of chemical mutation or radiation, and rate is 1 into 10 raised to minus 3. Now, if you see that particular history, so how induced mutation studies have been initiated in worldwide. So first report of X-ray was reported by Muller on Drosophila for the first time, and later in the next year, 1928, Stadler reported effect of X-ray on barley and maize. So that was the initiation of how mutations can be induced in any individual, maybe plant or any insect and so on. After the end of World War II, Auerbach and her team has reported use of chemicals. So initially only radiations were used. Later, chemicals were also used for induction of mutation. That was reported by Auerbach and her team. Now, in case of this particular mutation breeding, so how we can do this particular thing? So whenever any particular change is not, so whatever desirable change we want to create in a particular organism, so if mutation is not there, then we can do that particular conventional method, hybridization. But in that case, we may also observe some undesired characters. So considering that particular problem, can we have other option where we can keep all those positive characters and we can induce variability in which we are interested. So in that case, by the help of chemical or physical mutagen, we can induce heritable changes, and those changes will come at gene level, and because of which we can observe new and heritable characters. And hello, can you mute other participant? Hello.
बनसोडे सर, वन क्लिक मध्ये सगळ्यांना हे करा,
उनने वीडियो
सगळ्यांना
नीचे
सगळ्यांना
सरकार
म्यूट
का
करा.
कंपटीशन
वो कह रहा है कि जाति का वो कह रहा है कि आजकल पकड़ वाला चौदह का
Hello. Can you hear me? Hello.
हा सर, बोला.
yes,
तुमचा आवाज येतोय, बोला.
No. In that case, when we are inducing that particular mutation, so that will result into creation of heritable variation, and those variation should be heritable one. And based upon that, we can find out, or we can select a particular variety which is showing establishment of those heritable changes generation after generation. So that forms a particular science that is known as mutation breeding. So in which we are changing the morphology of plant, or we are observing the maturity, where we are observing early maturity, or showing stress resistance, that is biotic and abiotic resistance compared with the parent. So those characters are induced in that particular mutation. Now, if we want to induce that particular mutation in any individual, so I'm concerning with only plant. So what we have to do, we have to first find out that particular material. Selection of material is very important. Then which kind of mutation we want to use for induction of mutation, that we have to find out. Then how we have to give that particular treatment, that also we need to understand. And after giving treatment, we have to find out how we have to handle the particular material. It may be seed propagating, or it may be clonal propagating. So that we have to find out, and then accordingly we have to start the so selection of material. Now, in that selection of material, uh, first step is very important. So, which kind of variation we want to create? So, nature of variation that we want to induce, that we have to first think, and accordingly we have to choose that particular mutagen. So, for which we have to first find out some earlier literature or earlier workers' results. We have to find out, and accordingly, then we have to select that particular material as well as a particular mutation. Now, during selection of material, whether that plant is propagating through seed or through propagule and so on, that we have to consider. In case of in vivo and in vitro, if we want to induce mutation, then we have to induce first that particular callus or embryo and so on, and then we have to do that particular induction of mutation. Now, when we have to do that particular induced mutation study, so in that case, it is observed that a variety is very popular, but which is showing some deficiency in one or other character. Say, for example, it is showing susceptibility to biotic stress or abiotic stress, or showing lodging one, or having long maturity period, and so on. So, in that case, can we improve that particular variety without disturbing other positive characters? So for that purpose, we have to first find out what are the lacunae in that popular variety and obtain its particular seed. So seed must be pure one, and then we have to start that particular treatment. So while selection of material, we can have that particular option of selection of seeds if plant is propagating with seed, or if we want to induce that particular hybridization program, then these pollen grains are collected, these are irradiated, and then these are involved in that hybridization program, or vegetative propagules or cones or bulbs or even complete plant can be can be used for induction of mutation. So in that complete plant, where that gamma garden facility, so where we are keeping that particular plant in field in that gamma garden, and slow dose is given continuously for 8 or 10 days. So in which we are trying to find out how that particular plant material is showing effect in live plant. So in this case, choice of mutagen. So we have to again find out what kind of particular change we want to carry out in that particular plant. So induction of desired trait. So accordingly, we have to select that particular mutagen, whether we want that particular large alteration or any specific change we want in that particular plant phenotype. So accordingly, we have to use that particular mutagen. So if you see that particular radiations, so different kinds of radiations are there. So mostly ionizing radiations are used. Non-ionizing radiation, that is UV, is used only when pollen grains or when we are using that microbes and so on. So for exposure of that one, these are using. But on large scale, these ionizing radiations are used, and these ionizing radiations will do different effects. So which will carry out chain of reactions, and this chain of reaction will induce several kinds of mutations. So in case of chemical mutagens, so where we are observing that how these particular chemicals are getting attached with that particular genetic structure, that is DNA, ethyl or methyl group from alkylating agents, and then they show that particular degradation of that particular base. And lastly, because of that particular degradation, a basic site is created, and that will show mispair or recombinogenic changes will observe during replication, and during next replication cycle, these changes will get removed, and then we observe that a transition or transversion. So such kind of different mutagens are there from different groups, that azide group, hydroxylamine, and so on. So where we are observing deamination or distortion of DNA double strand, or transition or tautomerization, and so on. Now, once we understood which kind of mutagen we have to use, whether radiation or chemical mutagen, then actually treatment will start. How we have to give the treatment. Now, as far as chemical mutagen is concerned, if you want to give the treatment, then three parts. It is divided into three steps. So pre-soaking, actual treatment, and post-soaking. So pre-soaking. So in which what we are doing, we are keeping the particular seeds, whatever healthy, dry, uniform seeds which we have collected from that particular variety, we are keeping in water so that imbibition will take place, or in some cases we are using some solutions which will enhance the effect of mutagen, in which we are giving that particular treatment. That is known as pre-treatment. So it is showing that time from 6 to 8 hours or even 12 hours. So we have to imbibe that particular seed, and then we have to actually give the treatment of particular mutation with variable concentration. So for which we have to find out that particular LD50, so lethal dose that we have to find out. And after giving that particular treatment, we have to put that particular seed in running tap water, or we have to give constant shaking during the treatment. And after treatment, we have to again wash that particular seed. Purpose to remove the excess of mutagen before sowing that particular seed into the soil. So in that case, whenever chemical mutagen or radiation we are using for induction of mutation, it is very important to find out LD50. So someone is saying that it is called as a pilot experiment where we are trying to find out how our treatment is showing that seed germination percent. In that case, it should be less than 50 or near to 50. Then such kind of concentrations are used. Concentration which is killing all the individual, or concentration which is not killing any individual, such kind of concentration are not used. So lethal, sublethal, subvital, and vital. So such kind of observation we are observing. So we have to find out LD50 of radiation or chemical mutation, and accordingly we have to start that particular mutation. Now, this is the particular flow chart how we have to do that particular mutation. So take a particular mutation, different concentrations, we have to prepare and obtain that particular seed material. We have to imbibe that particular seed and give the treatment. Then after treatment, we have to sow that particular seed in M1. So just like in case of conventional breeding, we are observing F1, F2, F3, and so on. So in case of mutation, we are calling the, or we are naming the generation M1, M2, M3, and so on. In M1 generation, we are not getting any kind of mutant, but here we are trying to find out what kind of sector, chlorophyll deficient sectors are there. So those chlorophyll deficient sectors we are trying to observe. Then we are also trying to find out what kind of abnormalities are there, phenotypic abnormality or injuries induced. Then your chromosomal abnormation at the whatever root tips we have fixed. Then we are trying to find out pollen sterility and so on. And those plants which are showing that particular chimera, they show 100% induction of mutation. So we have to concentrate on them in next generation. So at the end of M1 generation, we have to harvest those plants individually, and in M2 generation, we have to sow those seed on plant to row basis or plant to progeny basis. And in M2 generation, we have to go for rigorous selection. Why we have to say that rigorous selection? Because in this generation only we are able to find out maximum variability with respect to different traits, and whatever our trait of interest is there, that we have to select in that M2 generation, and then we have to harvest those plants individually. So after harvesting these M2 plants individually, then we have to go for M3 and M4 again plant to row basis, and then we have to find out whether these are showing a constant expression of character, or whatever trait we have selected, it is heritable one or not. Later, these after completion of M4, they are showing the homogeneity. Then we have to go for seed multiplication, or further evaluation is there, or somewhere these particular mutants are used for hybridization. So at the end of M7, M8 multi-location trial, then it is ready to release that particular variety as a mutant variety. So in which it is helping us to reduce the time period compared with the conventional breeding. So this is earlier phase was related to in vivo. So in vitro also we can induce mutation. So what we have to do? So what kind of change we want to induce, whether we want to induce a change at the biochemical level or phenotypic level and so on. So accordingly, first we have to select the plant bird or the explant. Then that explant we have to culture it on the medium and allow to produce that callus and shoots. And then at the level of callus or at the level of shoot, we have to give that particular treatment of radiation, or even we can give that particular chemical mutagen during that level. And then we have to find out the variation, and such kind of somatic variations we have to screen, and those somatic variation we have to find out whether these are showing variation induced at first generation, that is MV1, MV2, MV3, and so on. So that we have to observe, and later we can confirm it whether whatever trait we have obtained, whether it is heritable one or so. Now, after induction of mutation, we can find out how the changes are observed there. So based upon that particular mutation types, we may observe broadly three types of changes. So intergenic or point mutation. So where a particular sequence of particular gene, as we know that base pair ATGC will get changed. It may be transition or transversion, and then it will lead to that particular variation in that particular trait. Or intergenic or structural mutation, where we may observe breakage of chromosome, and these chromosomes will get portion or entire chromosome will get deleted, or it will get duplicated, inversion or translocation, and so on. Or last one, where chromosome number is getting changed. So such kind of changes we may observe. And lastly, whatever because of this intragenic or that particular chromosome number changes, we observe two types of changes in that particular individual in field, that is under macromutations and micromutations. So macromutations are those kind of mutations which are visibly observed, and these are controlled by few genes, whereas micromutations, where we are not able to observe the individuals visually, where we are taking the observations, and then we are by applying statistics, we can find out how these individuals they are showing variation compared with the parents. So macromutations and micromutations we are observing, and then we have to find out whether these are true breedings and so on. Now, once we obtain a particular mutant through chemical mutation or radiation, then what we can do? So that mutant which we obtain from this breeding program, mutation breeding program, so that can be used as an improved crop variety. So after confirmation showing true breedingness, we can release that crop as a variety which is improved one, or we can induce that particular male sterility or create additional genetic variability, means that trait is not present in that particular germplasm stock at that particular crop, and we are interested in that one. So what is the option? We have to find out that particular addition of that particular trait from unrelated, taxonomically unrelated crop, and then we have to do the breeding program and so on. So it may take much time, and expertise also required. So instead of that, we have to find out that particular mutation, induced mutation option, where we can create that additional genetic variability, and after observing its particular performance, stable inheritance performance, then we can release this one. Or it can be used to show that improve adaptability. So if that particular crop is showing adaptability to any abiotic stress, so which is not previously reported in that one, so that we can create and we can introduce that improved crop in the field. So there are certain examples. So where how these particular different crops in which mutants have been developed and released, which are showing resistance to different biotic stresses, such as disease resistance against bacteria, fungi, mycoplasma, viruses, even parasitic plants also. So against them, these particular crops, the mutant varieties have been developed and released. In case of abiotic stress, so some examples are there. So where we are trying to find out how this particular stress against drought or salinity, or that particular cold one, or high altitude, and so on. So different abiotic stress, whenever these are there, so ultimately productivity of crop is getting reduced. So in that case, we can have that option of development of abiotic stress resistant plant through induced mutation, and then we can release this. Again, apart from this biotic and abiotic stress, we are also have some examples. We are having examples where we observe that some crops they get matured earlier. So if you see some examples where that crop is getting matured by 30 to 45 days in case of rice or soybean, by 1 month, or even in castor by 150 days. So what is the benefit of this one? So crop, if it gets mature early, so it will help the farmers. If the crop is from Kharif season, then if it is getting matured earlier one, so that field will get empty early, and the farmer can go or can have a second crop in his field in Rabi season. So that is the benefit of this one. Then grain yield. So it is observed that because of these particular mutants, enhancement in yield is observed. So that is a positive point of this one. Then again, grain quality. So there are certain examples. So how that particular mutants, they are showing that enhancement in grain quality, fatty acid profile is there, or protein content is there, or seed size is, or seed color, and so on. So these are the different examples. Again, there are certain examples where in these crops, wheat, barley, rice, green gram, black gram, where these mutants were used as one of the breeding parent, and conventional breeding was carried out, and from that segregation population, different lines were obtained, and these lines they are showing superiority over parent as well as the mutant one. So ultimately, our aim is to provide improved cultivar to the farmers, so that farmers will get maximum yield and will not show any kind of problem as far as controlling the pathogen, growth of pathogen, or plant are not showing that adaptability to different abiotic or biotic stresses. Now, if you see that particular impact, global impact of this particular mutant. So if you this particular slide shows showing that particular how different mutants from different crops, when these were released worldwide, so how they have changed the entire agriculture as well as economics. So if you see the example from India, that rice one, so just because of release of the PNR 102 and PNR 381 mutant, so 1,700 million US dollar annual crop value has increased. So just because of release of that one mutant, how farmers are getting benefit. So these are the examples from different parts of the world. So again, in case of black gram Maharashtra, so this TAU, this black gram mutant was released in 90s, where because of introduction of this particular mutant, how that 64.7 million US dollar production has been increased just because of release of one mutant. So how mutants, they are helping improve cultivar, they are helping farmers for enhancement of their particular yield. These are the different varieties which are released in India. So if you see that particular table, so what are the varieties which are present at the end of that particular crop, which are released from 2000 to 2010, and earlier varieties are there which are older one and which are released by different institutes from India. So rice, castor, and so on and so forth. So these are the different varieties which are showing superiority over the parent, and not only that, they are showing enhancement in yield, they are showing resistance to different biotic and abiotic stresses, then they are showing improved nutrition quality, and so on. So just I want to give some example. So when that particular black gram TAU, so this TAU variety was released. So before releasing, the area under cultivation was 400, that yield 419 kg per hectare yield was there. But later, because of introduction in 1999, we observed that there is enhancement in yield per hectare, that is by 212 kg per hectare. So ultimately, if you see that particular 2 billion annual income, 2.5 billion annual income has increased, and compared with just investment of 1 crore rupees. So how one mutant, if it is developed and it is released, so it can change entire agricultural system, and it will help or boost that particular economy of that particular farm. So if you see that particular data from IIA, so in which it is observed that around 3,200 mutant varieties were released worldwide, and among them cereals, they are having maximum ratio that amount, followed by ornamental legume plants and so on. So where we observed that these particular mutants release worldwide, Asian country, in which India, China, Japan, Korea, Pakistan. So these are showing major contribution as far as induction of mutants in cereal, pulses, vegetable, and so on. Afterward, Asia, Europe is there, which is producing high number of mutant varieties. Now, this is one part, how mutants are developed and how these mutants have changed the entire agriculture pattern or the cultivation of that particular crop in that particular country or in particular area of that particular country. Now, how mutants are used, how that radiation treatments are useful for crop rotation, second part. Now, in that second part, we will see during storage, we observed that because of storage pest, our crop is getting damaged very on large scale, and ultimately we are suffering. So, can we protect our crop? So, in that case, it is observed that yes, there are certain methods where sterile insect technique (SIT), so which is used for pest control. So, what we have to do? We have to use that particular insect. Uh, uh, so, hello.
Hello.
Hello, Ranjit.
हाँ सर एक मिनट म्यूट ऑल.
Hello.
हाँ सर बोला.
Okay. Now, in that case, this particular radiation is used. So where we are using that particular insect and we are exposing that particular insect for radiation. So here, so those insect larvae we have to expose for radiation, and later these particular larvae will hatch and will produce that particular male one. And because of this particular radiation, large alterations will take place in that genome of that particular organism, and ultimately because of this one, some insects, so insect will become sterile, so male insects. So these male insects are then sprayed, and what is the purpose or what we are expecting here? So these male insect will meet with the wild female, and because of this, what will happen? Whatever female will lay eggs, so all these eggs will not hatch and will produce larvae. So ultimately, we are controlling the birth of this particular insect or that life cycle of that particular insect. So sterile insect technique. So in which what we are doing, we are trying to minimize the incidence of that particular growth of insect, and major achievements are where 30 different states, they are using this particular method for exposure of a particular insect for radiation, and then they are releasing this particular insects so that they will come in contact with the wild flies, and they show the fertilization. Ultimately, there is no growth. So because of just spraying these particular insect, how that economy has increased in case of chili.
मीटिंग बंद कर.
So it will lead to that particular enhancement in yield, and all those crops will get protected from the effect of insect. The food and environment protection. Now, in this case, so when after harvesting, these particular storage pests will cause that particular damage to these stored food grains. So in that case, can we protect our stored food grains or even vegetable fruits or fresh? Yes, these end products, these are exposed to radiation with low doses. And what is the purpose? Purpose is that whatever microorganisms are there, so these microorganisms which will get, and ultimately shelf life of that particular end product will get enhanced. So where gamma rays or electron beam or X-rays, these are used for exposure, and ultimately we are trying to protect that particular plant from the exposure. So these are the particular steps where we are exposing that particular food material. It may be your flesh one, or seeds, or fruits, or vegetables, or spices, and so on. So that will ensure the food hygiene. It will give the safety to the food. Then it will overcome the quarantine barriers. So we know that during quarantine, if whatever export or import of material is there, and if it is fine showing presence of microorganism or insects and so on, or weed seeds, then we are rejecting that consignment. So if that if any consignment we are exporting or importing, if it is free from that particular growth of microbes, then it will be readily accepted by that country. So in that case, we can use this particular option of exposing that food grains or that end product by radiations, and it will be protected by protected from the microorganisms. So these are the different examples how that particular doses were used. So slow doses or minimum doses used for exposure, and after exposure, these particular plant products or even frozen sea products or the deflation and so on. So they will get protected from the microorganism. So next one, application of food radiation. So in which it is observed that about 60 countries, they are already applying this particular protection of food grains by exposing with the radiation, and because of this they are reducing the loss in yield which is observed because of that particular microorganism which are growing during the storage condition. So this is about how mutations are induced, then how mutants derived from this particular different program, how they have changed the agriculture, then how mutations are or radiations are used for protection of plant from insect or food grains. These are protected from the microorganism during storage and so on. So this is the half portion which I want to explain how mutations are helpful for crop improvement and crop protection. Now, I will give some of my self experiences from last 5-6 years, which you are doing work on different crops, and how mutations are helpful for induction of variability and improvement of a particular crop plant. Now, so we are working on some rice land races from this particular Kolhapur area. So these particular land races, which are so this particular project, Azaragansa, a non-basmati aromatic rice land race. So it is a land race which is growing in particular one tahsil of Kolhapur district, that is Azra, near Amboli, and that particular cultivar is famous because of its particular aroma. It is growing in that particular area from long time. It is having good cooking quality, good aroma is there, then expansion of or elongation, the grain elongation after cooking is a good one, and this particular genotype is adapted in that particular area, and for different purposes, this particular or festivals and so on. So this rice is preferred in that particular area, but there are certain challenges are also there. Okay, it is having good kind of aroma, and because of availability of high rainfall, relatively low temperature, fertile lateritic soil, and cool and dry condition during formation of seed and aroma retention. So all these conditions are prevailed there. But in spite of this, this particular cultivar is not cultivated. Why? Because it is having long maturity period. Then rainfall is there. If the rainfall is not or lateritic soil, so if this condition, if there is any change, then it will affect on the aroma production. Then yield is very less compared to the national check varieties. Then plant height is very high. So because of which lodging is a common problem, less number of tillers. So ultimately yield is less, lodging is there, and if you provide inorganic fertilizer, then again vegetative growth will be very, very fast one, and again lodging will be there. So in spite of all these particular challenges, some farmers, they are they were cultivating this particular cultivar in that area for their own urge, but they require improved cultivar, and considering this particular point, we have initiated this particular work, keeping in mind that how we can develop that particular high yielding, lodging resistant, DOF, and early maturing lines of Azaragansa. So in that, we have used that particular pure seed of Azaragansa, and we have used 3 mutagen, that is 2 chemical mutagen, ethyl methyl sulfonate, sodium azide, and radiation. So we have used 3 different concentration and doses, and these seeds were treated, and Then M1 generation. So in M1 generation, so already I have explained how we have to find out that particular trait. So in M1 generation, we have tried to find out what kind of changes are observed in case of chlorophyll deficient sectors, chromosome abnormality, pollen sterility, and plant sterile. So where these particular treatments, they are showing variation in that particular different parameters, lethality, pollen sterility, chromosome abnormality, and so on. So that we have to record. So this is a particular field view. So how that pollen sterility is there in that pollen. Then these are the chlorophyll deficient sectors which are observed during M1 generation. So these are the perfect indicators. Yes, these are the plants. They are showing that particular induction of mutation. Then we have to concentrate on this one. And at the end of M1 generation, then we have to harvest this plant. So these are the some chromosomal abnormalities, sticky metaphase, scatter metaphase, anaphase breach, and so on. So these were reported at chromosomal. So that indicates, yes, mutation has arisen at the chromosome level. So at the end of M1 generation, we have to harvest this plant individually, and then we have to sow this plant in M2 generation to raise the M2 population on plant to row basis. And where we are observing again chlorophyll deficient mutants. So these mutants will also indicate that, yes, what kind of mutation frequency is there, and from that particular mutation frequency we can find out effectiveness and efficiency of that particular mutagen in that particular cultivar. So these are the particular mutants. So albina, xantha, varieties, and chlorina. So these particular mutants we are observing at the M2 generation. So these will indicate that how mutation has arisen in that particular. So these are the step-by-step event, right from field preparation to the harvesting one. So how we are doing this particular work. And then in this case, in M2 generation, we should have maximum population. So look at this particular population. So from this one, we have to select that particular desired plant. So in this case, these particular chlorophyll mutants we have obtained. So different frequency of albina, xantha, chlorina, and sectoral mutants we have reported from this one. Now, during selection of this particular desired mutants, we have to find out the traits or some characters. So we have to consider. So for which purpose or on which basis we are going to distinguish desired plant from the undesired one or compared with the parent. So these characters we have considered during screening of plants from that particular M2 population, and we observed. So just bear with me because some font size is very smaller one. So in this case, we observed that how that particular dwarf mutants were reported. So dwarf mutants, so compared with the plant height, if we observe that 30 to 40% reduction in plant height, so that is variability. Then in case of other EMS mutant, EMS treatment, we observed that particular 100 centimeter reduction in plant height was observed compared with the control. So such kind of variations were observed. Then some of the high tiller number mutants where we are observing that particular enhancement in tiller number as well as effective tiller number compared with the parent, and we observed that there is enhancement in yield, which is twofold or even threefold yield is there compared with the control. So these are the particular high yielding mutant which we observed in M2 generation. So these are the dwarf mutants. So look at this particular dwarf mutant. So individual plant we observed. So this particular plant is showing variation compared with the other plants. So these are the particular dwarf plants. So which are showing altered phenotype compared with the plant. So high number tiller. So how the tiller number has increased. So individual plant by plant, we have to visit and we have to find out what kind of variation is there. Then in case of other treatment, we observed again there is 50% reduction in plant height is there. Then again maturity. So compared to 160 days, we observed there is a 25 days early maturity compared with the control, and among them some plants they are showing yield, but that yield is less than particular control. So but variation is there as far as maturity point of view is concerned. So these are the some plants which shows that particular early maturity from EMS treatment. In case of sodium azide treatment, we observed that there are certain plants which shows enhancement in tiller number compared with the control, and because of enhancement in effective tillers, we observed that there is enhancement in yield, which is around 3 to 4 fold yield compared with the control. Along with this, we also observed that certain plants which shows early maturity, which is 23 days, they get mature early compared with the control, and in which some plant they again show reduction in plant height. So these are the particular plants which shows high number of tillers and effective tillers. Same one. Now, in case of gamma rays, so in case of gamma rays, so surprisingly, we also observed that compared with the chemical mutation, we observed that these particular dwarf plants were there, as well as there are certain plants which are showing high number of tillers compared with the control. And if you see that particular yield, so it is observed that 3 to 5-fold enhancement in yield is observed. So this is very interesting one. So all these plants were harvested at the end of M2 generation. So these are the photographs which shows that particular high yielding plants, dwarf plants. Now, one thing is, so in that again during that some plants we observed which were showing lodging resistance. So at the time of harvesting, some entire control plant they show lodging one, but some plants they stand erect and they show that particular erectness till maturity. So these particular plants were also observed which are showing erectness. So if you see the neighboring field, entire lodging is there. So such kind of variations we observed. Now these particular plants were observed in M3 generation. So when we sown these particular plants in M3 generation, so almost the character has been retained there. So if you see that particular darkness, so that character has retained, and along with this particular enhancement in yield is also there. So these are the particular dwarf line which shows entire that true biddingness. So look at this particular line. So remaining plants they show tallness, and this particular line is showing that particular dwarfness. So again, this particular dwarf line. Yeah. So these are the individual plants which we have taken photograph. So in case of other plants, so this maturity, early maturity. So where again these plants they show early maturity, and again they are showing enhancement in yield. So whatever trait we have observed in M2 generation, so same trait is showing heritable expression in M3 generation. So these are the particular plants. We show this one. Then in case of other treatments of same Azragansa, so where we observed that early maturity is there, and along with this particular high yielding, look at it. So whatever mutants which we have selected in M2 generation, so they show 2-fold, 3-fold enhancement in yield was observed. So ultimately it is showing enhancement in yield and showing true biddingness. So this particular trait was observed in M3 generation. Again, similar that lodging resistant plants which we have selected, so they are showing lodging resistance in M3 generation, as well as they are showing enhancement in yield compared with the control. So look at this one. So entire that particular lodging remaining plants they are showing lodging one, and this particular line is showing that particular lodging resistance. So these particular plants were reported from M3 generation. So by observing its performance in successive M3, M4, M5, we have to find out how these plants they are showing that particular true biddingness. And once we observe that particular true biddingness, then we have to go for multi-location trials, and then after multi-location trial at the end of M8, M7, M8, we can go for that releasing of that particular variety, or we observe that particular stable inheritance, then that particular plant can be used as one of the breeding parent to start a breeding program. And from that particular breeding program, whatever segregating population is there, we can again find out a most desired one, and that plant again we can select, and then go for multiplication trial, and then we can release. So by one or other way, this particular end product of mutation breeding program can be used for improvement of that particular crop plant. So these are the experiences from Azragansa. So another cultivar is also there. So in that another cultivar which is Kajirka. So this particular Kajirka cultivar. So again, why we have selected these two cultivars? So basically, these two cultivars are cultivated in that Azra area of Kolhapur district. These are having good aroma. then these are showing adaptability in that particular area. If we are transplanting this particular plant in other area, or we are taking cultivation, no doubt they will give yield, but aroma is not produced, or that quality of aroma will be lesser, minimum one. With that, uh, another thing is that this particular Azragansa and Kajirga, they are nutritious one, means they are having high iron content or ferrous content is there. And during our field survey or consultation with the farmers, what we have observed that this particular rice cultivar Azragansa and Kajirga, these two rice cultivars are offered to the old age patients or mothers, lactating mothers. So because of consumption of this one, that iron content, your HB will get increase, and it will help to recover that particular old age patient or those who are not suffering from different disorder. So for enhancing their particular health, these particular cultivars are offered. So these particular cultivars are having some challenges, and those challenges we want to overcome by using that particular mutation building program. So next cultivar, that is landrace, that is Karajirga. Again, this particular project is sanctioned from DST SCRB. So objective was again to improve that Karajirga for yield, maturity, then your lodging resistance, and darkness. So this particular cultivar is grown in Kolhapur district. It is having typical aroma production, though production is less, but because of having that particular aroma and taste, this cultivar is grown and protected farmers for their own consumption from last several years. So challenges were there. So this particular again, Kalajirga is requiring 150 to 160 days period for maturity. Then it requires high rainfall. Production is very less compared to the national chick varieties. Then plant height is very high, and it shows lodging one. And ultimately, when that particular plant is having less tiller number, ultimately it will show lower yield. So and negative response is there whenever we are giving that inorganic fertilizer. So height will increase, and ultimately again it will show that particular lodging. Lodging is a common problem. So considering these particular challenges, farmers, they are facing challenges, and they are reluctant to take, in spite of having this particular good land traces which are showing acclimatization in that particular area. They are not ready to take because they are getting very less yield, which is not fulfilling that requirement, and then they are finding out other option of having hybrid varieties. But some farmers, they were interested in cultivating this one, but they require that particular improved cultivar. So considering this one, we have started that particular mutation breeding program in Kalajirga, and again we have used ethyl methyl sulfonate, sodium azide, and gamma rays, and we have given treatment with 3 different concentration and doses, and then we have completed the sowing. And during M1 generation, again we observed that particular polar sterility, chromosome abnormality, chlorophyll deficient sector, lethality, and in those characters we are recorded, which indicates that how mutation has occurred in that particular plant. So this is the particular field. So all this work we have carried out at Azra, though we are at Kolhapur, but these crops, they are commonly cultivated in that Azra area. So we have tried to observe all our experimental results at Azra location only. Then and then only we can. So we thought that then and then only we may get the expected results at Azra location. So we have carried out all the experiment at Azra. Now, so these are the particular chromosomal abnormality. So where these are showing sticky metaphase, that laggard precocious movement, anaphase bridge, and so on. So such kind of chromosomal abnormality indicates that yes, mutation has arisen in that particular M1 generation. So again, chlorophyll deficient sectors, these were reported in the M1 generation, which are the indicators of induction of mutation. So at the end of M1 generation, we have harvested this plant individually, and then in M2 generation, seeds were sown on plant to row basis so as to raise the M2 generation. So during again M2 generation, we have reported different chlorophyll deficient mutants, and so these are the views of how that particular field work step by step we have carried out. In this, look at this one. So population of M2 generation compared to Azra, it was less, but around 47,500 seedlings were there. So among them, we have reported that particular mutation frequency by observing the chlorophyll deficient mutant, albina, xantha, chlorina, and sectorial one. And these particular mutants, so that mutation frequency is used to find out efficiency and effectiveness of that particular mutation. So in M2 generation, again, these traits were used to find out how this plant they are showing variability compared with the parent. Now, in case of Kala, so compared with the Azraghansa, Kala, it has shown more height, and in M1, M2 generation, we observed that there were certain plants which are showing reduction in plant height, which are showing reduction in maturity period. So compared with the control, that days to maturity has reduced by 10 days. Then here, in case of plant height, height has reduced by 30 to 40% compared with the control. And if you see that particular yield per plant, though these particular early maturity plants we observed here, so their tiller number, effective tiller number, they were less, they were showing enhancement in yield. So certain mutants were there which are having high tiller number, so which are showing twofold enhancement in yield compared with the control. In this one, we observed that threefold enhancement in yield in that particular mutant compared with the control in case of EMS treatment. So these are the particular plants which are showing enhancement in tiller number. So some are dwarf where. So in case of gamma rays treatment, so what we observed that certain plants. So look at this one, 20 centimeter compared with 140 centimeter. So 20 centimeter height was there, but unfortunately that plant didn't produce fertile panicles. Very few seeds we observed, and in M3 generation, these seeds were unable to germinate. But other dwarf mutant, if you see in that dwarf mutant, so there is a decrease in 100 centimeter height compared with the control. And if you see that particular tiller number, again, those tillers are greater in number compared with the control. So such kind of different variability we observed in gamma rays. So look at this particular dwarf mutant. So this one, so which are showing drastic reduction in plant height, and again there is enhancement in tiller number was also observed. In case of sodium azide treatment, we observed that 50% reduction in plant height was there. Then there is enhancement in tiller number, effective tiller number, and increase in yield per plant was also observed. So all these particular plants were selected from this particular M2 generation from Kala and harvested individually at the end of M2 generation. So these are the pictures which shows how these particular mutants they are showing enhancement in tiller number compared with the control. In case of gamma rays treatment, now when we obtain these particular plants from M2 generation, again at the end of M2 generation we have harvested individually, and in M3 generation again we have sown this particular plant on plant to row progeny, and where we observe that, look at this particular plant. So plant height compared with control, 55 centimeter height is there, and if you see that particular yield per plant, it has showing enhancement in yield. So that particular change is also generation to generation, and what kind of environmental conditions are there, we may show variation in control, and along with that, so how our mutants they are showing enhancement in yield. So that so whatever character which we have observed in M2 generation, it is showing true breedingness in M3 generation. So look at that particular early maturity. So in case of early maturity, that particular plant is getting matured by 7 to 8 days, and so on. So these are the particular traits which we have observed in M2 generation. So look at that particular entire dwarf line, which is showing that particular expression of stable inheritance character. So this one. So dwarf mutants were reported in that particular M3 generation. So these particular observations were reported early maturity. So 8 to 10 days early maturity is very important as far as farmers field is concerned and farmers requirements are concerned. If we provide early maturating variety to the farmers, so farmers will get benefit. So in certain area it is observed that late month or that November, December, in that month, return monsoon is also there. So that return monsoon, if that crop is there in field and monsoon is there, so again, huge damage will be there. So if our crop is getting mature early by 8 to 10 days, and so farmers, they can harvest that plant early, as well as that field will get empty, and if based upon availability of water and resources, they can have second crop. So these are the benefits of that particular early maturity high yielding mutant for as far as farmer community is concerned. So as far as conclusion from our studies are concerned, so in this case we can say that how that particular genetic variability is there. So genetic variability can be induced through induced mutation. One, then if you want to induce that particular mutation, selection of mutagen is very important. So if you are using any mutagen, so its concentration or radiation, its dose, we have to choose first. So for which we have to go for that LD50, that lethal dose we have to find out, and then we have to go for that induction of mutation. In case of radiation, we can't go for that pilot experiments, and then we have what we have to do. We have to find out the early literature survey, and accordingly we have to select that particular doses, and then we have to start that particular breeding program. So from our study, we can say that moderate concentration, not lower concentration or not higher concentration, are useful. So moderate concentration of EMS or gamma rays or sodium azide, these were able to induce maximum desirable mutants from the present study. And compared with the Azragansa and Kajirga, if you compare this one, so it is observed that these particular variety, they show differential response, means whatever effects we observed in case of Azragansa, same kind of effects we have not reported in case of Kajirga, and vice versa from same mutagen treatment or radiation treatment. So that difference may be there. So that difference we have to note. So I would like to thank the different funding agencies, so which have funded us from last 4 or 5 years. So BRNS, then ACRB, DST, SCRB, then UGC, they have given us some just amount of funds to execute this particular work. So whenever we want to carry out a particular work, so it requires strength, and my strength. So these are the research students which are working with me, and at present, and some of the students, they have left. So at initial period of that particular project, these were with me, and later they left. And now these students, they are pursuing their particular study, and we are now in a position to release these particular mutant for multi-location trial. So now we are taking M5 generation this year. So M3, M4, we have confirmed this. Yes, these are the particular mutants. They are showing heritability, they are showing provenance, and at the end of M5 generation, we are trying to go for that particular multi-location trial, so that we'll able to release these particular improved lines for the farmers, and because of which farmer will get benefit. So whatever their particular requirement was there when we started that particular program, so farmers were expecting that yes, when we get that particular improved cultivar with high yield or early maturity or dwarf one, so we are reaching near to that particular objective. And again, from next 2 to 3 years, we will go for that particular multi-location trial, and at the end of multi-location trial, we'll have that particular option to provide that particular improved cultivar to the farmers. So with this, I would like to thankful to the organizer for giving me this opportunity to share my views about induced mutation in crop improvement and crop rotation. Thank you. Hello. Hello.
Hello. Hello, Gaikwad sir.
Yes, please.
Thank you very much, Nikhil Gaikwad sir, for enlightening all the delegates. I would like to request some of the participants to ask any question or query regarding his excellent research work. Anyone from participant? Okay. I think your presentation was so excellent, and so therefore it was so well explained that there was no query from the delegate side. Now the last thing it is vote of thanks. I would like to thank Dr. Nikhil Gaikwad sir, who is also a friend of mine, and he is also guiding us for the excellent research. He has given his valuable time for this webinar, as well as I would like to thank organizing secretary of this webinar, Dr. Ranjit Bansode sir, who has given tremendous effort for success of this webinar. I would also like to thank our college management as well as our principal, Dr. Narendra Tendulkar sir, for encouraging for this organization of not only this seminar, but also all other activities which are organized by Department of Botany. And last but not least, I would like to thank all the delegates who have attended this webinar from all over the country as well as from abroad also. I apologize for anything that was not working, such that many participants were interested to join the webinar, but due to constraint, we have allowed only 100 participants on the Zoom, and the rest of the participants have attended the webinar on YouTube link. So thank you one and all. I would also like to request all the participants to fill up the feedback form. The link will be shared on the WhatsApp group. So link will be open till 9 o'clock today. So please fill the form so that you can get the certificate. And lastly, also thank you one and all. Thank you, guys.
Thank you. Thank you.
Thank you.
पाऊणला सुरू झाली. एक मिनिट बंद करूया.